Chapter 3, Part 5, Biological Macromolecules. Proteins continued. Proteins can fold and unfold when they interact with their environment because they are molecules of amino acids that are charged, positive and negatively charged portions. They also have some unique characteristics that allow them to interact with the environment and change with changes in pH, acidity, and alkalinity, as well as changes in temperature. There are other factors that can, in fact, impinge or affect protein shape, but we're just going to focus on pH and temperature effects. Whenever we've changed the structure of a protein due to pH or temperature, we call this denaturation. A change in the structure of the protein can often lead to a change in the function of that protein. When a protein has undergone denaturation, we say that protein is denatured. Denaturation can be either reversible or irreversible depending upon the protein. If we take a look at the effect of temperature on protein structure and function, here we have an example of two chemicals or two proteins in the form of biological catalysts that we call enzymes. We have glucose 6-phosphate enzyme from a bacterium that lives inside of a human being, and we also have glucose 6-phosphate enzymes from bacteria that live in a hot spring. Notice that we have two different curves of protein function or enzyme function. We call these curves essentially the functional curves, or the relative activity of the enzyme. And notice that the relative activity of the enzyme is plotted in percentages on the y-axis of this graph compared to the temperature changes that we've exposed the protein enzyme to. We have a temperature minimum, which is on the left for both curves, a temperature optimum, which is the highest peak for both curves, and a temperature maximum, which is on the right-hand side. Notice that we have a gentle sloping or gentle increase in enzyme activity as we're going from temperature minimum to temperature optimum for both enzymes. Both enzymes at a lower temperature have sort of a maybe almost no activity or half activity in terms of relative activity. We get to a temperature optimum, and that's where we see our greatest, most rapid activity of that enzyme. As we increase the temperature to temperature maximum, for some enzymes, we actually wind up with zero activity. This is an example of a denatured protein that's completely lost its function when we get down to zero activity. For the hot springs enzyme, derived from the hot spring bacteria, when we reach a temperature maximum, we still have activity occurring. So we'd actually have to heat this protein extremely high, far beyond boiling water temperatures, perhaps, to completely irreversibly denature that enzyme. So for temperature, most proteins, not just enzymes, but proteins in general, can have a temperature minimum, where they have very little function, temperature optimum, which is the ideal temperature, and a temperature maximum, which is essentially way too hot. For pH, we can see a similar type of graphic distribution of function based on distribution of pH. Here we have two dotted curves of chitinase activity. So chitinase is an enzyme from bacteria that either live in acidic pools, which is the gray line or graph, or we have chitinase from soil-dwelling bacteria, which is in blue. Notice for both chitinases, similar to what we saw for temperature, we have a minimum, which is on the left-hand side. We have an optimum, which is the ideal pH for the best sort of ideal function of that enzyme. And then we have a pH maximum, where we get too extreme in terms of pH for these enzymes to function properly. We again plot pH on the x-axis versus relative activity of the enzyme on the y-axis, and can see that as we go from the pH minimum to the optimum to the pH maximum, for each enzyme from these two different organisms, these minimum, maximum, optimum pH are going to be different, but we still see a similar type of distribution or curve from each one. So enzymes from different organisms function best at different pH and temperature based on how the organism itself has adapted to those different environments. And in fact, it's this flexibility or adaptation of these proteins that have allowed these organisms to live in these different environments. So protein denaturation can be affected by pH and temperature, also what's called ionic strength, in other words, what kind of salts or other chemicals are surrounding it, the solubility of that protein in the aqueous environment in which it's found, and for some proteins, we call the normal protein structure. Once they've gone through denaturation or loss of biological activity, it remains that way. It's irreversibly denatured. For different kinds of proteins, depending upon, again, the type or where it's found, we can see that some denatured proteins can actually undergo renaturation and regain their activity again. Every organism is unique, every protein is distinctive, so we can't make any broad general characteristics of proteins or groups of proteins. We have to look at each individual one, study them to figure out if they're irreversibly or reversibly denatured under certain conditions. Here's an example of an animation that shows us a little bit more about protein denaturation and why once you cook an egg, you can't take that egg that's been cooked, put it back in the refrigerator, and expect it to turn back into the jelly or the gel, clear gel that you first saw when you put the egg into the skillet. You might need to have Flash active on your computer when you're running this animation if you do so using the PowerPoint that's separate from this video and audio.
An egg provides a good example of how heat can change the structure of proteins. Proteins are found in both the egg white and yolk, and these proteins have different structures in raw versus cooked eggs. Let's consider the proteins in an egg white. In their native uncooked state, the proteins are chains of amino acids that fold into complex shapes. Several types of bonds form between amino acids in a chain, stabilizing the structure of the protein. When the egg is cooked, the heat makes the molecules vibrate. The bonds linking the amino acids in chains are strong covalent bonds that withstand the heat. However, most of the bonds that stabilize the three-dimensional structure of the protein are weak bonds, typically hydrogen bonds, which break, allowing the proteins to unravel. When two unfolded proteins make contact, they form new bonds with each other. A large net of interconnected proteins forms, and the egg hardens. Light can no longer penetrate the egg, which is why the clear portion of the egg turns white.